She uncovered the secret to cellular immortality and changed how we understand aging, cancer, and life itself. This is the story of Elizabeth Blackburn, the Nobel Prize winning scientist who cracked the code at the very ends of our DNA. Elizabeth Helen Blackburn was born on 26 November 1948 in Hobart, Tasmania, Australia, the second of 7 children in a family where both parents were medical doctors. Her mother, a practicing general practitioner, and her father, a physician and hospital administrator, provided a household steeped in the values of science, medicine, and public service. Growing up in such a medically oriented environment, Blackburn developed an early appreciation for biology and human health. As a child, she was captivated by nature and the structure of living things, often collecting and examining insects and small animals. Books like Darwin's On the Origin of Species sparked her imagination, and she would later cite this early exposure to evolutionary theory as a formative influence. Her natural curiosity, paired with a rigorous academic upbringing, made science a natural path. When the family relocated to Melbourne, Elizabeth attended University High School, where she thrived academically and developed a growing passion for chemistry and biology. Encouraged by her teachers and family, she pursued biochemistry at the University of Melbourne, completing a Bachelor of Science degree in 1970 and a Master of Science in 1972. Her postgraduate work focused on biochemical processes at the cellular level, laying the foundation for her future in molecular biology. Eager to deepen her expertise, Blackburn moved to the United Kingdom to begin her PhD at the University of Cambridge, one of the world's premier scientific institutions. There, she worked in the prestigious Laboratory of Molecular Biology under the mentorship of Fred Sanger, a future 2-time Nobel laureate and pioneer of DNA sequencing. During her time at Cambridge, Blackburn investigated bacteriophage DNA, exploring how viruses infect bacteria and replicate their genetic material. The research honed her skills in molecular techniques and DNA analysis, preparing her for the revolutionary work that would follow. She completed her PhD in 1975, emerging as a young scientist with both intellectual depth and hands-on experience in cutting-edge research. In 1975, Blackburn accepted a postdoctoral fellowship at Yale University, where she joined the lab of Joseph Gall, a respected cell biologist. At Yale, Blackburn began researching the structure of chromosomes using the ciliated protozoan Tetrahymena thermophila, an unusual organism with many small chromosomes, each with distinctive DNA ends. Her breakthrough came when she discovered that the ends of chromosomes, known as telomeres, contained repetitive, non-coding DNA sequences. These sequences protected chromosomes during cell division, preventing essential genetic material from being lost. This discovery challenged the prevailing notion that the ends of DNA strands were either insignificant or simply degraded over time. Blackburn's work would go on to transform the understanding of cellular aging and genome stability. In 1984, while at the University of California, Berkeley, and working with graduate student Carol W. Grider, Blackburn identified an enzyme that could replenish telomeres, telomerase. This enzyme added DNA repeats to chromosome ends, acting like a cellular clock reset. Their discovery was a landmark in molecular biology, illuminating mechanisms behind aging, cancer, and cell immortality. After Yale, Blackburn accepted a faculty position at UC Berkeley, where she continued her telomere research. In 1990, she moved to the University of California, San Francisco, where she became a professor of biochemistry and biophysics, and eventually directed the UCSF Helen Diller Family Comprehensive Cancer Center. At UCSF, Blackburn's lab deepened the investigation of telomerase in cancer cells, where unusually high levels of the enzyme allow cells to divide indefinitely, one of cancer's hallmarks. Her team demonstrated that manipulating telomerase activity could potentially control the proliferation of cancer cells and slow cellular aging. She published extensively, mentored future leaders in science, and became a prominent figure in the international research community. Her work found applications in regenerative medicine, diagnostics, and aging-related disorders, placing her at the intersection of basic science and clinical innovation. In 2002, Blackburn was appointed to the President's Council on Bioethics by US President George W. Bush. She used the platform to advocate for the responsible use of science in public policy, particularly in support of stem cell research. The dismissal ignited widespread protest within the scientific community. Blackburn emerged as a public advocate for scientific integrity, evidence-based policy-making, and freedom in research, arguing that political interference in science threatened innovation and public trust. She co-authored articles in major journals warning against censorship and bias in scientific advisory bodies, and she inspired researchers to engage more openly in public discourse. In 2009, Blackburn, along with Carol W. Greider and Jack W. Szostak, was awarded the Nobel Prize in Physiology or Medicine for the discovery of how chromosomes are protected by telomeres and the enzyme telomerase. The Nobel Committee hailed their work as one of the most important in the field of biology, highlighting its implications for understanding cancer, aging, and inherited diseases. Blackburn became the first Australian woman to win a Nobel Prize, marking a historic moment not only for her personally, but also for women in science globally. She received accolades from institutions around the world, including the Albert Lasker Award for Basic Medical Research in 2006, the Royal Medal of the Royal Society in 2010, Time magazine's 100 Most Influential People in 2007, induction into the U. In 2016, Blackburn was appointed president of the Salk Institute for Biological Studies, a prestigious research institution in La Jolla, California. During her tenure, she promoted interdisciplinary collaboration and diversity in science. She emphasized the importance of mentoring young scientists, particularly women and underrepresented groups, and advocated for science funding and climate awareness. Though she retired in 2018, Blackburn's legacy continues to grow. Her research remains foundational to studies on aging, cancer therapy, and longevity science. She continues to lecture, write, and advise organizations on global health, science education, and ethical innovation. Elizabeth Blackburn is married to John Sadat, a molecular biologist and fellow academic. They have one son. Despite spending much of her professional life in the United States, Blackburn remains proud of her Australian heritage and has maintained strong ties to the scientific community in Australia. She's also known for her humanistic values, humility, and dedication to service. Outside of her lab, she enjoys music, art, and nature, embodying the balance between intellectual rigor and emotional depth. Elizabeth Blackburn's career is a testament to how curiosity, perseverance, and integrity can reshape entire fields of knowledge. Her discovery of telomerase redefined our understanding of how cells age, why they sometimes become immortal, and how that process can be manipulated to treat disease. As a Nobel laureate, advocate, mentor, and trailblazer, she has inspired generations of scientists and thinkers. Her legacy is not just one of scientific discovery, but of using knowledge to serve humanity with compassion and conscience. From the quiet shores of Tasmania to the global stage of science, Elizabeth Blackburn's journey is a powerful reminder of where curiosity can lead. If you found her story inspiring, make sure to like, subscribe, and join us for more incredible biographies that uncover the mind shaping our world.